Thank you so much for the lovely introduction. And it's really the only silver lining for me of COVID-19 is to have the opportunity to present uh, to you, Professor Saxena, and your colleagues in India. And thank you very much for giving me the opportunity. So I thought I would do is tell you about our research at that intersection of chemistry, engineering, biology, and medicine. Okay, for some reason now, okay, there we go. So I like to describe our research as biology-inspired polymer design. So what we really try to do is try to answer unanswered questions in biology and solve problems in medicine. And this is our raison d'etre in a way for the work that we do. So we're very much interested in this idea of tissue mimetics and growing cells in a 3D hydrogel. So in an environment that mimics the way cells would grow natively in us. We're also very much interested in cell, drug, and protein delivery strategies, and invented this hydrogel of hyaluronan methylcellulose, which I'll tell you more about later in the talk. And also have done some work in targeted drug delivery with polymeric synthesis. So as was mentioned, our research is focused half on regenerative medicine, which is really in the central nervous system, in the spinal cord, in the brain, and in the eye for blindness. And the other half of our research is really focused on cancer. So what I thought I would do for you this evening is just tell you about some of the highlights of our research. And I know that you have the opportunity to ask questions online. And so I encourage you to do that, to ask questions throughout the talk, and then I'll definitely try and answer them at the end of the talk. So I like to think about our research as really this idea of shaping the future of medicine and really approaching these fundamental challenges and problems that we've been faced in society for decades or hundreds of years, but really have no way of overcoming them. And so this idea of making change and approaching these problems differently is what really excites me about our research. And so looking at that intersection, so bringing our knowledge in chemistry and engineering together with that of our colleagues and collaborators in biology and medicine is what's very exciting to me and really drives our research. So I'll tell you three stories. The first one relates to personalized medicine and cancer. And the second two stories will relate to regenerative medicine in blindness and in stroke. So while we're very focused on solving problems in medicine, our research always starts with a fundamental question and a I wonder if question. And in the early 2000s, we had one of those I wonder if questions, which was I wonder if we can grow nerve cells in a 3D environment. So we and many other people could create these beautiful neural circuits on 2D flat surfaces. And we wondered if we could do the same thing in 3D. Could we guide nerve cell growth in a three-dimensional matrix, just like we guide it on flat 2D surfaces? And the reason why we were interested in asking this question is because, of course, if we were trying to mimic the way cells grow natively in us, then we really should grow them in a three-dimensional environment because we, of course, are 3D. And if we were trying to mimic the way cells would grow natively in us, then we should probably think about not just the dimensionality, but also the mechanical and chemical properties. And so, growing cells on hard plastic dishes are not particularly biomimetic. If you think about most of our tissues are soft, some are elastomeric. Of course, bone is hard, but all the other tissues and organs in our body are much softer. And so again, growing cells in a hard plastic dish is probably not a great way to mimic the way they grow natively in us. So that was that simple question that we started with. And then over the past decade, we've taken that simple question, I wonder if, and built greater complexity into our hydrogels and gone from asking that simple question I wonder if we can grow cells in 3D to now using those cells that we're growing in 3D for drug screening and drug discovery. And the picture of the people that you see below are actually not even everybody, but many of the people who've worked in this area for the past decade or so. So the way we grow cells normally are on flat tissue culture polystyrene dishes. We might coat them with a protein, with polylysine, a polypeptide, or laminin or collagen, but still the underlying surface is very hard. And most of what we've learned from cell biology is by growing them in these hard plastic dishes. And so I don't want to underplay the importance of growing cells in these 2D culture wells, because we've learned an enormous amount of biology by doing that. But I also can't help but wonder how much biology we've missed by growing cells in these flat 2D hard plastic dishes. So the work that we've been doing is focused on growing cells in hydrogels. So hydrogels, as you know, are just water swollen materials. And so, well, Jello, hopefully you have Jello in India as well. We certainly have a lot of Jello here that kids love to eat. I certainly wouldn't want you to walk away from my lecture thinking that we're doing research in Jello, but I just show that as an example of a water swollen material that many people are familiar with. So we're not using Jello, but we're working with hyaluronic acid. Now, hyaluronic acid or hyaluronan turns out to be a material that's expressed throughout our bodies. We're interested in using it because it's overexpressed in many cancers in the body. And so it seemed to be a good starting material if we're trying to emulate the extracellular microenvironment or the extracellular matrix in which cells would grow. It seemed like a good idea to start with one that was already overexpressed in that environment. Hyaluronic acid has been used for many medical applications. It's been injected into knees for osteoarthritis. It's being injected into faces to get rid of wrinkles, and it's now a very common additive to many creams. As I said, we were interested in hyaluronic acid because it's overexpressed in cancers. And but if we wanted to make a material with any mechanical properties, we'd have to figure out a way to cross-link it. So unlike Jello, if you've ever made it, you just put it in the refrigerator and it forms a gel, so a temperature-sensitive gel. Hyaluronic acid, we would have to chemically cross-link it to form a hydrogel. So some of the early work we did in growing cells in a 3D environment was done by Ying Lo. And Ying had that first question of, I wonder if we can guide the growth of primary dorsoroot ganglion neurons in a three-dimensional hydrogel. And so what she did is she chemically modified this agarose hydrogel with the RGD peptide. And she put a fluorescent tag on the RGD peptide. So that's what you can see in green. And then she cultured primary dorsoroot ganglion neurons on top of the gel. And she found that those cells and axons were able to migrate through the gel. And those are what you see in red and orange. And what was really interesting about working with this material is because we were interested in guiding cell growth in 3D, we knew it was important to start off with a material that itself was non-adhesive to cells. So we have an inherently non-adhesive gel. And then by chemically patterning in the RGD peptide, and we used a laser to do some of this chemistry, or patterning, we're able to create a volume in which only the cells would grow, because it's almost like they had a fence on either side and they could only grow in that cell adhesive path. Well, a couple of years later, Yuki Aizawa was wondered if we could differentiate neural stem cells to oligodendrocytes by immobilizing a growth factor, platelet-derived growth factor AA, onto these hydrogels using similar chemistry into oligodendrocytes. So typically PDGF would be added as a soluble factor to neural stem cells and promote their differentiation to oligodendrocytes. But Yuki wondered whether we could immobilize that factor and get the same type of differentiation. And that's what you can see here with this RIP positive staining, showing that the cells do in fact preferentially differentiate to oligodendrocytes. Well, one of the limitations that we saw with some of the work that Ying had done and Yuki had done is that we were really limited to growing cells in 1D. So if you think about it, even though the cells were growing down into our three-dimensional hydrogel, they're only growing in one direction. And so when Jordan Wozniak joined the lab, we wondered whether we could get those cells, or whether we could advance our photochemistry and our laser technology to do 3D chemical patterning. And that's what you saw with that spiral. So the spiral isn't particularly useful from a biology perspective, but it did demonstrate that we could have control in the X, Y, and Z directions using multiphoton lasers, and now using a multiphoton labile protecting group. And the group that we used was a bromohydroxy coumarin protecting group. Well, similarly, what you see here in these U of Ts representing the University of Toronto is our capacity to have a very precise control of the chemical patterning of our hydrogel. And so this is just using a red fluorescent molecule. And the reason I show you this is because I'm hopeful that you can see 4 layers of U of T. I'm not sure if you can see my pointer, but you'll see for sure the first layer, that's very bright, and the second layer, which is a little bit less bright, a third layer, and maybe you can see a 4th layer as well of a U of T. And the reason I show you this is because not only could we have precise control in 3D of where we were chemically patterning this hydrogel, but we had precise control of how much of the fluorescent molecule, in this case, was being patterned. And so by controlling the exposure to light, the first one has greater exposure. This one has less, and progressively less exposure to the multiphoton laser, and therefore less of the fluorescent tag was immobilized. And so what this told us then is that we could use this chemistry to create gradients. And gradients, of course, are very important for guiding cell growth. So this work was then further advanced by Ryan Wiley. So coumarin itself is fluorescent. And so what Ryan was really interested in doing was asking the question of whether he could immobilize 2 proteins, so not just a fluorescent tag now, but 2 proteins simultaneously in a hydrogel. And so he immobilized ciliary neurotrophic factor, CNTF, and sonic hedgehog simultaneously with this precise 3D control. Basically, what Ryan did is he took an agarose hydrogel and he modified it with a bromohydroxy coumarin protected thiol, and then cleaved off that bromohydroxy coumarin, leaving these reactive thiol groups. And then he reacted those thiol groups either with a maleimide streptavidin or a maleimide Barnase, and then could immobilize each of the corresponding proteins modified either with a streptavidin that would link to the biotin or a bar star that would link to the Barnase. And in this way, he was able to immobilize two proteins shown in red and green here. And you can see, again, with that precise three-dimensional control of where those proteins are. Now, this shape, you might wonder, why did Ryan immobilize such an unusual shape? And the reason is, and I don't know if you guys can tell what this shape looks like, but it's meant to look like an eye. So, where you would have the lens and the globe shape of the eye. And the reason why we were interested in that shape generally was we were interested in seeing if we could preferentially differentiate stem cells, retinal stem cells, into the different cell types that make up the retina. And that's why we were using CNTF and sonic hedgehog, because they were known to be important in the differentiation pathway. So fast forward now a number of years, when Sean Owen joined my lab as a postdoctoral fellow, he wondered whether we could take some of these fundamental questions and apply them to cancer. And so the question was, could we now grow cancer cells in a 3D environment and then use that for more predictive drug screening and perhaps even drug discovery? And again, you might say, well, why would you want to do that? And the reason is that, as I mentioned earlier, 2D cell culture is pretty fast, but we lose a lot of information just by the inherent limitations of growing cells in 2D and on hard plastic dishes. Our gold standard for screening is xenografts. So that would be, taking biopsies of human cancers and growing them in immunocompromised mice, and then screening drugs that way. And so while the predictive power is very high there, it's very time consuming and costly. And often patients can't wait and will start their treatment regimen while the xenograft model is being advanced. And so the hydrogel, or this 3D model, is kind of in the sweet spot. It should give you more predictive power than what we have with 2D culture, and should be less expensive in terms of time and cost than the xenograft model. And so this is an area that we focused on, and there are other materials. There's a large body of work of people working in this area, but a lot of those hydrogels haven't been validated. And by what I mean by that is that even though you can grow cells in a 3D matrix, we don't actually know if what we're seeing is representative of what would happen natively in us. And so that's been a big focus of our research. So this work was led by Roger Tam, and what Roger Tam did was a head-to-head comparison of growing cells in 2D tissue culture polystyrene, which you see on the left, or growing them in our 3D hydrogels. And I'll tell you more about these Hyaluronan hydrogels that we worked with. The great advantage of working with a 3D hydrogel is that we can look for not just how toxic a drug is, but also how it impacts cell invasion. And you can imagine, for a highly invasive disease like brain cancer, glioblastoma, or lung cancer, any highly invasive disease, being able to screen a drug for not just cell viability, but cell invasion, we thought was very important. And this is additional information that you can only get in a 3D gel or a 3D environment. So as I mentioned earlier, we moved from agarose, which was very well defined and enabled us to do some really interesting chemistry and biology, but we moved from that to hyaluronan because it's much more relevant. And Roger was working with a rare lung disease called lymphangioliomyomatosis. And that disease is in the lung. It's smooth muscle cells invade into the lung, and there's an overexpression of hyaluronan in that system. So hyaluronan is a natural ligand for CD44 and CD44 variant 6, which was important in the invasive phenotype of these cells. And as I mentioned, we can control the chemical and mechanical properties through cross-linking. So we used an MMP degradable cross-linker. This would allow cells to invade by actively degrading the hydrogel. Cells will migrate through different mechanisms, and another mechanism in which they'll migrate through tissue is just sort of by an amoeboid mechanism where they're pushing their way through. And so we're able to mimic that in the gel by incorporating some physical crosslinks with methylcellulose. And methylcellulose will form physical crosslinks. And then also cell adhesive peptides were incorporated into the hydrogel by inclusion of vitronectin peptides. And these vitronectin peptides were shown, we did some screening on the cells to show that they express the integrin receptors for those peptides. So I have just this one slide for the chemists in the audience. I understand there's a lot of mixed backgrounds of people here today, which is wonderful. If you're a chemist, you can look at all the chemical structures. And if you're not a chemist, you can look at just these different colored lines, which I'll show you, and I'll guide you through the synthesis of this biomimetic hydrogel. So we start on the left with hyaluronan, and then we introduce a furan group through chemical modification of the hyaluronan. This then allows us, this furan group allows us to do Diels-Alder chemistry. This is cross-linked with that MMP degradable peptide cross-linker with a bismulamide functionalized peptide. Again, if you're not a chemist, you can just look down at the diagram on the left and this is color-coded. So you can see that we've now cross-linked those blue lines with red lines. And we've introduced those vitronectin peptides, again, using the same Diels-Alder chemistry, except only using a monofunctional vitronectin mimetic peptide, and link that also to the hyaluronan hydrogel. The next thing we wanted to do was introduce those physical crosslinks so that cells could migrate through the gels by different mechanisms. And so this required us to chemically modify the methylcellulose hydrogel in order to chemically immobilize it into the gel. And so what we did then is we introduced a thiol group, so this SH group shown in pink, onto the methylcellulose. And then the methylcellulose could then be linked to some of the, just through a Michael-type addition, through some of the unreacted maleimide functional groups. Now, the methylcellulose, as I mentioned, is there to form physical crosslinks, and that's through just through the hydrophobic, very weak hydrophobic interactions between the methoxy groups or the methyl groups on methylcellulose. So this hydrogel, it had a pretty low modulus of about 1 kilopascal, which is similar to normal lung, human lung tissue, which has a reported modulus between about 1 and 5 kilopascals, but obviously much closer than what we see with conventional 2D cell culture, where the modulus is about 3 gigapascals. So this work was done in collaboration with Bill Stanford's lab at the University of Ottawa. And what we're really interested in doing is not just taking this one gel at a time, which is often what we have to do in 3D hydrogels, but see if there's a way that we could automate this drug screening. Because if we're going to try and actually screen hundreds of drugs, so not tens of thousands, but at least hundreds, we needed some sort of automated system. So we worked with a 384 well format. We labeled the top of the gel with silica beads because, as you can imagine, there was a little meniscus, and we wanted, if we were going to look for cell invasion, we had to make sure that we were actually looking at cells invading into the hydrogel and not ones that were simply falling into that meniscus. So we labeled the top of the gel with these silica beads, and we stained the cells with Cytox green. And then using a 384 well format, we could then use a high content image analysis system. We used the Celomics image analysis system, and then wrote a script to allow us to look for cell viability and cell invasion into these gels. And so this is something that enabled us to screen a number of different drugs at the same time. So there's a lot of data on this slide, but this work has been published. So if you want to focus on it more, I welcome you to do that. But what you can see in this heat map, and here what we did is we looked at 80 different genes, 80 different drugs, and then looked at those drugs to see what target pathways they were pursuing. And at the same time, what we did is we did a heat map. So the drugs and target pathways are what you see on the left and right of this heat map. So the drugs are all on the left and the targeting pathways are all on the right, and those are the colors underneath target pathways. And what you see in the heat map in terms of green and red is green means that we saw a drug that reduced cell invasion and reduced cell viability. And the red is one that did the opposite. So increased cell invasion and viability. And this is all relative to controls. So what's really interesting to us in this heat map was, I didn't tell you much about the disease, this Lamb disease that we were targeting, but most of the drugs that are being pursued in clinical trials are going after the mTOR pathway, which is, you can see in red on the side of the heat map. That's the pathway on the right-hand side. And what you can see is some of the drugs going after the mTOR pathway are actually pretty low on our heat map and don't do that much for targeting invasion or viability. Some are closer to the top, but they're not our top hits. And so what we can see now by looking for viability and invasion in our screening tools is that this opens up new drugs and new pathways that we probably wouldn't have thought about previously. And so this is an area that we're quite excited about and are pursuing actively. So I'll just end this story by saying we've taken this to investigate brain cancer and lung cancer. So this is lung cancer, and what you see in the first two rows is the top view, and what you see in the last row is the side view. And the first three columns are all different types of primary lung cancers. The next two are two cancer cell lines. And the one on the far right is actually a healthy bronchial epithelial cell. And what you can see with that one is that it really doesn't. So this is R, the far right on the bottom right. What you can see with that is those cells really don't invade into the hydrogel. So they survive on it, but they don't grow into it. Whereas all the lung cancer cells grow into it. Now they grow in by different mechanisms and to different extents, which is just an opportunity for us to do further research on this. But what was really important for us was that the healthy cells don't invade. And so this then, it gives us greater comfort that this is a good model to study disease and lung cancer cells. This is just work that Alexandra Baker did by taking the work we've done in those 3D hydrogels and now actually growing cells in a 3D environment. So instead of just plating them on top and watching them invade, we can now grow cells within a 3D environment, and that was really based on designing some new hyaluronan oxime hydrogels. And so what you see on the left are these breast cancer spheroids that have formed over several weeks, and what you saw on the right was just a 3D rendering of cells growing in that 3D matrix. So this was the longest of those three stories, but what we saw was that we could create these 3D hydrogels that mimic the native extracellular microenvironment, and that we can use them to start doing drug screening and hopefully some drug discovery. We are extending this now to brain cancer and lung cancer and breast cancer, and working with different organizations to study their drugs. Oh, I don't think you can hear that. Is that right?
No, we cannot hear that.
Okay, I'm sorry. I'm going to go on. That was my wishful thinking. Okay, so let me go on now and tell you about two different stories. These stories will be shorter. I'm going to tell you about our work in regenerative medicine. So there's a couple of fundamental challenges that we face in regenerative medicine. And one is cell survival and one is cell integration. So there's two different ways that we think about regenerative medicine. One is in cell transplantation, and that work I'll tell you about in the context of our work in blindness. And the other is stimulating the stem cells that are already resident in us. And that's the work we do in the context of stroke. And so I'll tell you that work. But whether you're transplanting cells or stimulating resident stem cells or endogenous stem cells, those challenges of cell survival and cell integration are paramount and are really, I would say, limiting the field. And so these are areas that we're very focused on. So I want to introduce you now to this hydrogel. Actually, I talked about this at the very beginning. This is a very simple physical blend of two materials, hyaluronan and methylcellulose. So we talked quite a bit about hyaluronan already when I was introducing you to chemically cross-linking it. This now is just a physical gel. Methylcellulose is inverse thermogelling, so you heat it up and it forms a gel, and hyaluronan is shear thinning. And what hyaluronan does is it basically lowers the gelation temperature of methylcellulose. And so this is a very simple video, there's no sound in it, of our hyaluronan methylcellulose hydrogel. Now we've colored it blue just so you can see it. And so for any of the engineers in the audience, this is a really cool engineering material because it's basically a gel in our syringe, and we apply a little pressure and it flows, so it's shear thinning. But as soon as that pressure is released, it forms a gel again. And the size of the blue blob is not important. It's more important just that as soon as the gel comes out of the syringe needle, it forms a gel again. And that's just a physical gel. And what I was trying to show you is that this can go through a very fine needle. It can go through a needle that would go into the back of the eye of a mouse, so through a very fine 34 gauge needle. So in the context of blindness, there's age-related macular degeneration, which results in central vision loss, retinitis pigmentosa, which results in peripheral vision loss, and a diabetic retinopathy, which results in a dappled vision. And so while we have drugs that will slow the progression of this disease, what we don't have is a way to stop disease progression or reverse it. And that's really the promise of regenerative medicine. So this is work that we did with and continue to do with Derek van der Koy's lab. His lab discovered that we all have retinal stem cells. They're found in the ciliary body in the eye. And those cells have the capacity to become all the cell types that make up the retina. So you know, we see because light comes into the eye and hits the back of the eye, which is our retina, these cone and rod-shaped cells, these colored cells that you see at the back of the eye, those are the photoreceptors. Those are the cells responsible for vision. And they have a very intimate relationship with these cells, the retinal pigment epithelial cells. And Brian Balios is an ophthalmologist now, but he was a PhD student in our lab, and he looked at the delivery of photoreceptor cells derived from these retinal stem cells to the back of the eye. And we knew, and most people still today, inject cells just in saline. And so we were quite interested in delivering cells in this hydrogel of hyaluronan methylcellulose because we had shown that it would promote greater cell survival and distribution in the eye. So I'm not going to go through all those details, but happy to refer you to this paper that was published in Stem Cell Reports. And maybe I'll just take you to the highlight, which is we were working in blind mice. So these are genetic blind mice, which means obviously that they can't see. And if you shine a very bright light at the eye of a blind mouse, one of these blind mouse, the dilation area won't change. You know, if you go out into very bright light, or if you're in a dark room, your pupils will be fully dilated, and then if you go to a bright light, they'll shrink. So what you can see is we expose the mouse's eye to a very high dose of light, and in red you see that their dilated area doesn't really change. But when we injected mice with cells in our hyaluronan methylcellulose hydrogel and with a pro-integration factor, we found that the dilated area started to decrease, and wherever you see a star, it was significantly lower. And so this was quite exciting for us. But at the same time, if a normal mouse had such a bright light shined on its eyes, that dilated area would have gone down to about 15 to 20%. And so while this went down to about 85%, we recognize that there's still further for us to go. So we've been looking at a number of different ways to promote greater cell survival. And one area that we looked at was the co-transplantation of retinal pigment epithelial cells, so these gray cuboidal cells, with these photoreceptors. And this was, or it's just about to be published in Biomaterials, so this is really hot off the press. And, you know, I'm recognizing I know that, do we still have an 11 or whatever
Yeah.
time it would be?
Yeah.
Do we have enough time, or I want to make sure there's time for questions? Well, so you'll just tell me when you want me to stop.
Sure,
okay.
no problem. Go ahead.
Okay, thank you. So in this study then, what Nick Matrusas did is he wanted to see if there was any benefit of transplanting those retinal pigment epithelial cells, those gray cuboidal cells, with the photoreceptor cells. And he did a visual acuity test where basically you look, it's called optokinetic head tracking, and you're seeing how the eyes track with this screen of black and white lines, and the black and white lines get closer and closer together. And what he found was that when he delivered, so this, when he did these analyses, he didn't know which sample he was analyzing. But afterwards, what we realized was the best visual acuity was achieved by those mice that had those, that's the green line, where we saw both photoreceptors and RPE transplanted. And that was, we only saw significant benefit with both of those transplanted, and we didn't see that significant benefit with any of the others, even though we did see some visual acuity returning with the RPE itself. Oops, that's weird. So the other study he did was a functional study about a light avoidance assay. And that is, as you might know, mice are nocturnal, so they prefer to be in a dark room. And if they have any vision, they'll be more active and spend more time in a dark room than a light room. And so we wondered if we would see that difference as well. And what's really interesting, so if I draw your attention to the far right, to the wild type animal, you can see that that animal is more active in the dark chamber, which is the dark line, versus the lit chamber, which is what we'd expect. But the other thing that was really interesting is that we only saw that significant difference, again, when we co-transplanted the photoreceptors with the RPE. And we didn't see a significant difference in any of the other controls. And so the reason why this is happening, we believe, is that each cell, the transplantation of both cell types is helping the other one survive better, and maybe also helping some of the endogenous tissues survive better as well. So that's hopeful, but still we're only getting about 10% of vision recovery. And so we're actively engaged in looking at delivering cells in not just this hydrogel, but a hydrogel that has growth factors or pro-integration factors that will further promote their survival. And we've developed an affinity controlled release system to achieve that. And so basically what this means is that we can deliver proteins in hydrogels with cells without incorporating additional polymers. And by simply controlling that on-off interaction between a protein and its peptide binding partner, so shown here between green would be the peptide immobilized on our hydrogel, and yellow, that yellow dart, has an affinity for green. Then by controlling that affinity, we can control the release of red, which would be our therapeutic protein of interest. So, Professor Saxena, should I tell this last story, or would you like to stop?
Yeah, we have another 2-3 minutes. Yeah.
Okay, so I'll tell you this story very briefly, and I apologize for going through it so fast. This last story, it will be short, and it's focused on stroke. And what we're interested here is stimulating the endogenous stem cells that are already resident in the stroke brain to promote recovery, versus cell transplantation, which was the story I just told you. So one of our collaborators, Cindy Morset, has shown that by delivering 2 growth factors directly to the subventricular zone in the brain, we can stimulate the endogenous neural stem cells. But the technique they used was highly invasive. While I'm showing you what would look like a human brain, I just wanted to tell you that we're working with animal models of stroke and not actually human brain. But what you can see here is by inserting a cannula into the brain, you're going to damage brain tissue. And so we developed this less invasive hydrogel that you would apply, kind of like a patch that you put directly on the brain, and then have the molecules diffuse from that into the brain tissue. And so again, we used our hydrogel, and here we delivered 2 factors, erythropoietin and cyclosporin A. Erythropoietin to promote neurogenesis, and cyclosporin A to stimulate the endogenous stem cells. So I'll just skip to this data. This work was, oh, actually it says unpublished, I apologize. It was just published in 2020. And this is work that Anoop Tuladar did. And hopefully you can see that this is a rat that is in a cylinder. And in this assay, we're just looking at how it's using each of its paws. So if you have a stroke on the left side of your brain, it will affect your mobility on the right side. And so what we're doing is looking at how this rat is using that contralateral paw relative after stroke, relative to where it was pre-stroke, and with a series of different treatments. And so there's a lot of data here. I'll just draw your attention to the pink bars, because what we see in the pink bars is we see significant improvement when we deliver these two molecules, cyclosporin and erythropoietin, so C and E, versus just either erythropoietin, cyclosporin vehicle, or just injury alone. And we saw a significant difference at both day 10 and day 20. So we did another assay, which I won't go through, but we saw, again, improved functional repair with the co-delivery of these two factors. And when we tried to figure out why, one of the things we found was that we had greater tissue repair with the co-delivery of both factors versus each alone. And each factor seemed to be targeting a different pathway or a different mechanism to promote repair, which is why the two in combination was quite important. So I think I'll end there. I apologize for starting a little bit late and maybe going a little bit longer than I thought. I was able to tell you about our work in cancer and regenerative medicine. And I'm going to skip this slide. Oops, and I'll skip this slide too. I wanted just to get to, my question for all of you is how to change the world. We're trying to change it in science. We do a lot of outreach. I would have shown you some of these research to reality videos if they worked. But I'll end here by just acknowledging everybody in my lab and all of our collaborators, and thank you all for your attention.
Thank you very much, Professor Shoichet. It was really and indeed an eye-opener for all of us how all the three different branches of science and engineering have culminated together in your talk itself. And that has actually led to several questions. And I hope that I'll be able to do justice with all the questions in that case. So I'll take up questions one by one. So the first one is, was there any disadvantage of using the hyaluronan?
So I think one of the limitations with using hyaluronan in our cancer research, I think that's the question, is we can only make pretty soft materials. It's so if you really wanted to model bone or a harder material, then that would be a limitation. And I think if you wanted to model an elastomeric material, that is a limitation with that as well. So
okay.
Yeah.
Next one is there any effect or damage to the breast and the lactiferous ducts in common women due to the COVID-19 infection?
Oh, well, I'm really not an expert in COVID-19. Yeah, I really don't know the answer to that question.
Okay.
I apologize.
No problem. Does the cell transplantation help for cataract eye?
Oh, I don't think so. The cataracts are there's actually pretty good treatment for cataracts just in getting your lens replaced. And so the cell transplantation that we've been focused on is really in the back of the eye versus the cataract being at the front of your eye.
Okay. I'll take up the next one. Can you comment on the role of the chitosan-based materials in the regenerative biomedical research?
Yeah, chitosan is a really interesting material. We've used it before, and a lot of people use it. I think one of the challenges with chitosan is there's lots of different types of chitosan. So chitosan is derived from crustacean shells, and so obviously hyaluronan is a naturally derived material as well. And so with all naturally derived materials, we have to really make sure on purity and reproducibility. So chitosan has been shown to have some really interesting properties and has shown to have some not so interesting properties. And I think it really probably depends on the type of chitosan you're using. So it's an interesting material because it's positively charged. And so you can do some, some interesting protein and oligonucleotide delivery with it.
Okay, I'll take up the next one. Dr. Viggy, would like to know your insight about the application of decellularized extracellular matrix for cell seeding.
Yeah, so these are all excellent questions. So the decellularized matrices have shown great benefit in a number of different tissues in terms of regeneration. I'm thinking about some of the work that Steve Badalak has done in terms of digit regeneration. And I think it's a really interesting material to start with because, again, you're starting with something that's already present natively. And so there should be some advantages to that. You know, the disadvantage is it's not as perhaps precisely controlled in terms of the chemical structure. So, you know, our reason for using hyaluronan is that it's already overexpressed in cancer tissues, and so it's already expressed natively throughout the body. And so I think you could use the same argument for wanting to use the decellularized matrices. And they bring, I think they have some advantages and disadvantages. And the advantages is they bring a lot of that native structure already to what you're trying to do. And the disadvantages would just be whether it's always reproducible. But I think people have been able to overcome that and shown some really great benefit with it.
Okay, I'll take up the next one. Can you please explain the plasma process which enables to activate all surfaces of complex porous structures such as scaffolds to covalently attach biomolecules and hydrogels?
Yeah, so plasma is I'd say it's kind of like a brute force method to chemically modify. So it's used all the time, just even in tissue culture polystyrene. You know, those polystyrene dishes are typically modified by plasma modification, and so it's very effective. And I think it's also an effective way to get into a porous structure, as long as you can get it to penetrate into the pores. And I think as long as the pores aren't too small, then that works out pretty well as well. So basically you can use plasma to functionalize materials and then do chemistry on those functional groups. There's probably not that many functional groups that are present. But I think with efficient chemistry that works actually quite well. It's not an area that we have used that much, but I do think it is a very effective method to functionalize materials.
Okay. Uh, can biomimetic hydrogel be used to culture hematopoietic stem cells which differentiate into the blood cells?
Yeah, that's a really good question. We did some work with Peter Zanstra's lab a number of years ago, working with hematopoietic. Well, I'm not sure if we used hematopoietic stem cells, but I think so. Yeah. So, and the question is just what are you trying to mimic when you're growing them in that environment? So, you know, one of the things we've learned working in this area is that and it's obvious, but each cell type is different. So while the hydrogel, what we've designed are these platform technologies, we often have to tweak them a little in order to make them really useful for each of the different cell types. And so I would expect you'd have to do the same thing for the hematopoietic stem cells.
Okay. Uh, in the genetic blind mice, what changed after transplantation of the retinal stem cells and which cells got restored?
Yeah, okay, thanks so much for asking that question because I know I just completely rushed through my last two stories. So I told you about two, you know, I summarized probably like a decade worth of work in about 5 minutes. So what Brian Ballios had done is he had taken both human and mouse retinal stem cells and shown that he could differentiate them into photoreceptor cells. And these are the rod photoreceptor cells, which are responsible for night vision. And we injected those in our hammock hydrogel with a small molecule called alpha aminoadipic acid, which promotes integration of the cells once they're transplanted. So those cells were transplanted and resulted in that we saw enhanced survival and enhanced integration when we transplanted the photoreceptors. And then Nick Matrousis took primary photoreceptors from mice and human embryonic stem cell derived retinal pigment epithelial cells and co-cultured those or co-delivered those together into a different model of blindness. That was actually a degenerative model of blindness where he saw vision repair.
Okay. You have illustrated in one of the slides that the xenograft is difficult to study individual components of microenvironment. Is hydrogels are better substitute than xenograft despite of lacking the bioactivity?
Yeah, so nothing can replace the animal model of disease or humans, right? I mean, that's why we progressively test things in in vitro in animal models and then in people really through clinical trials. So I mean, a lot is lost in the 3D system. So I wasn't, but a lot is gained over the 2D system. And so that was really my message, that we can do a better job of screening things in vitro by working with a more biomimetic system, and then hopefully have greater success when we go into the animal models and into humans. And the other, great thing about in vitro is we can obviously work with human cells. And if we can work with human cells in a 3D environment and that environment is more mimetic, then hopefully we'll learn more about those drugs and how they would perform in vivo. But we're missing so much, like the blood supply, right? And so many other cell types. So we're starting to build some complexity into our hydrogels in terms of looking at multiple cell types. But still, that is it's very simple.
Okay. So there are plenty of questions. What I'll do is I'll take up the last three because it's already overshot our time itself. So I'll take up the third last. Uh, what is the time period required to invade the cells in the lung tissue model?
Oh yeah, that's a great question. So we wait about 3 or 4 days, or maybe 3 to 5 days. So it does take the cells some time to migrate in and invade.
Okay. When hydrogels are used for delivering proteins in brain, in case of stroke, how do you manage to target a particular site in the brain? Because the diffusion will be mostly random.
Yeah, so that's an excellent question, and you're absolutely right. We don't have a good way of targeting right now, is the short answer. So really, you're right. So I want to just mention that we put this directly on the brain, so not on the dura. So we have to drill a little hole in the skull and the dura and apply this directly on the rat brain. But the good news is we've achieved a way to get around what's called the blood-brain barrier, because we're delivering the molecules directly to the brain tissue. But then you're right, they just have to diffuse through the brain. So again, I wasn't able to show you all of the work that Anu Tuladar had done, but he was able to show that those drugs do diffuse through the brain to where the endogenous neural stem cells are. And so we know that they diffuse far enough, at least in a rodent brain. The challenge is how do we extend this to the human brain, which is a lot larger.
Okay. So I'll take up the last question. It has two parts. Could biomaterials mimicking blood-brain barriers and retinal blood barriers be created as model systems using these hydrogels?
Yeah, I think that's a great question. There's some really beautiful work on blood-brain barrier work, not using these hydrogels, and I think others are looking at using the hydrogels to create that. We had thought about doing some work in that area um, a number of years ago, and at least at that time, we thought that the models that people had using transwells was already pretty good, and that we didn't necessarily have to do that. But I do think that it is an interesting area, and pursuit. We are interested in endothelial cells and co-cultures with endothelial cells, and that's something, so not quite the blood-brain barrier, but seeing how those different cell types interact and inform each other.
Okay, I'll take up the last one now. Can this technique be used to heal the nerve damages in the case of Alzheimer or other memory-based cases caused due to nerve damage?
Yeah, so we did some work in Alzheimer's, but not in the context of regenerative medicine. And I think it's a great idea. And so the challenges are going to be the same, though, that some of the questions even highlighted. So how do you target those cells to regenerate? We know that our brains do have a certain amount of plasticity. So that means that we can rewire our brains. Obviously, it's easier to do younger than with our young brain versus an older brain. And you know, Alzheimer's disease typically affects us when we're older. But by discovering neural stem cells in the brain, in the adult brain, we know the brain does have the capacity to regenerate and to be plastic. So I don't know what those factors would be. And I think in doing that, we would have to be careful not to you know, just rewire the brain, because that could lead to epilepsy or you know, something negative. But I agree. I think it would be, that area is ripe for somebody to come in and explore. So that could be quite exciting.
Okay, thank you. Thank you very much. There have been several questions which I may have missed because of the paucity of time. I really would like to appreciate your patience of answering all the questions. And I would like to share that there have been a consistent 350 plus attendees all throughout your talk itself. So on behalf
Oh,
of the National Academy of Sciences, India, and the Upadhyaya College, I would like to thank you for sparing your valuable time and talking to us, sharing your work which you have been doing in your lab, and any concluding remarks from your side. Thereafter, I'll close the session.
okay. Well, thank you so much for giving me this wonderful opportunity to zoom into India. It's on my bucket list, so I really look forward to a time when we can travel again and I can meet you all in person. Thank you so much.
Sure, sure. We'll be more than glad to host your talk in person, maybe next year, anytime in your convenience itself. Thank you. Thank you very much, Professor Charlotte. Thank you.
Thank you. Bye.
Okay. Bye-bye. Bye-bye. So I'm closing this session, and I request all the attendees to kindly fill up the feedback form which I have shared in the chat section before leaving the meeting. Thank you very much.